There are four main ways in which ions can cross the cell membrane. First one being ligand-gated channels. These come in two types, the one where the ligand receptor is on the inside of the cell and one where it's on the outside of the cell. You then have voltage-gated channels which open at a specific voltage. There are stress-gated channels which open under certain stresses, so like actual stress pressure. And then there's some where there's no receptor involved, and these are called leaky Potassium channels or K+ channels, and all of these actually sit in the plasma membrane, and they have two conformations. Those are either open, which means that they're active, or closed, which means that they're inactive. These channels are very important in maintaining osmotic balance of cells because water can pass freely through the membrane via osmosis. So these things called sodium-potassium pumps, which are K+ and the M+, which is just at the top there. So when there's a low solute concentration outside of the cell, so like you've got not a lot of like salt molecules outside the cell, but a high solute concentration inside the cell, so you have a lot of salt molecules in the cell, it creates a lot of osmotic pressure outside. This means that water can then move into the cell by osmosis, causing swelling because it wants to dilute that high concentration of solutes. The issue is now that the cell is sort of really swelled and the pressure can cause it to actually burst. If we take a look at this example, you can see here that there's a high concentration of sodium ions extracellularly, so outside the cell, but a high concentration of potassium ions and proteins, which are negatively charged, inside the cell. So despite the fact that both sodium and potassium are positively charged, these negative proteins make the intracellular space more negative than the extracellular space, which means there is a concentration gradient. Because of this, the potassium ions try and move into the cell, while the sodium ions try and move out the cell. These are done via the channels that I was mentioning earlier. The first one being the lithium-potassium channel. This sort of doesn't have any sort of regulation to it. It just sort of like opens and closes as it wants to. Next are the sort of like creamy ones. These are the voltage-gated channels. You can see the first one is a sodium-gated one, and then the other one is a potassium-gated one. The voltage-gated sodium channel also only exists in neurons, unlike the leaky channel, which exists in all cells because it's very important for homeostasis. And then last but not least is the ATP-dependent sodium-potassium pump. These are important in maintaining the membrane potential and for signalling in neuron cells. This pump is very, very important in facilitating the transport back and forth of these sodium and potassium ions so that the charge differences stay the same, so that this diffusion cycle can keep happening. And to just summarise again, it is an electrochemical gradient that these ions move against or move to reduce. So if it's a very high negative concentration, then it'll try and move to the positive, and so forth in the reverse order. And then this is just a little table that I thought you might find useful. It's sort of got all the different transporters and channels on, where they're found, what sort of energy source they use to allow for this movement of ions, and just their function. So feel free to pause if you want to have a little look.